Right. Well, I want to thank everyone today for joining us with the IPIC webinar. My name is Dr. Laura Greiner. I'm currently the director here at the IPIC office. I have the joy of being the moderator today and introducing you to Dr. Josh Selsby. Dr. Selsby is a professor here in the Animal Science Department at Iowa State University. He received his BA degree from the College of Wooster in biology and his MA from The Ohio State University in exercise physiology. He followed that up with a PhD from the University of Florida, also in exercise physiology. And then he did a brief postdoc as well for the University of Pennsylvania. He came to Iowa State well, that's been almost 20 years ago, 16 years ago, I guess, back in 2008. And he joined the animal science team, and his focus has really been on the mechanism of muscle injury and the development of countermeasures for some time. So he's worked in a large, diversified area. A lot of his focus has been around muscular dystrophy and looking at different opportunities there. But another interest of Josh is that he's been focusing on has been around heat stress mediation in skeletal muscle. And so he's going to spend some time today going through some of that work with us and sharing with us some opportunities that it's in that realm for pigs. So with that, Josh, I'm going to turn the floor over to you.
Okay, well, thank you for that. Yes, my name is Josh, and you can, if you have questions and compliments, that is the email address to use. Other things, please use somebody else's email address. So I'm going to talk to you about some work today, and really this has been driven by a former graduate student, Tori Rudolph, investigating the impacts of heat on barrows and gilts. And where I'd like to begin is essentially here, just a consideration of how temperatures are changing. And what you're looking at is the globe, and what you're looking at are changes according to an average in a given region. So periods of red you'll see are above average, and blue is below an average. And what you'll see, and if you look at any one spot, you'll see it get red, and you'll see it get blue. But beginning in the 1980s, things get red and they stay red. And what you'll notice is that appears to be true in just about every place, particularly pig producing regions, including here in Iowa. And so all of that is to say it's hot, it's continuing to be hot, and it's likely to get hotter. Now, thermic injury exists on a spectrum. And when I got into this, I was really interested in this idea of therapeutic hypothermia. So we could actually use brief exposures to high intensity heat, and it actually caused increased protein synthesis and decreased muscle atrophy and did great things with a biomedical bend toward them. And there's actually some people now that are using those sorts of interventions to maintain insulin sensitivity to help offset some of the negative consequences of diabetes. And then on the other end, you have heat stroke, and this is what gets all the attention. This is actually a fatal condition. In some subpopulations, fatalities range upwards of 50%. So this is something that is catastrophic. But existing in the middle is this, what we term heat stress. So this is going to be longer term exposures, sometimes hours, often that extends to days, that elevate core temperatures in pigs by about 1.5 degrees to 2 degrees C. And this is what's really the culprit and the kind of the enemy of pig production in the summertime. And I think there's some cool data now also in the fall and early spring. So this is the story. Yes, heat stress, of course, is it has a negative impact, on agricultural economics in the United States. This is a $3 to $4 billion a year problem as we think about total animal agriculture. And then when we boil it down just to the pig component, it's somewhere in the neighborhood of $1 billion. I think the numbers now that we're using are around $900 million to $1 billion per year. Now, abatement is the biggest strategy we have to fight heat stress, and many of these are water dependent, which hasn't been a major issue until recently. And there's certainly talk now of water restrictions out west, and in Iowa, I think we just came out of a drought that had lasted quite a while. We have the infrastructure to do this. There's many places that don't. So I offer that just to say, Right now we're okay. Is this going to be a problem moving forward? Perhaps. The other thing that I want to highlight to you is that this is a problem now. Oftentimes, when people talk about heat stress and environmental change, etc., they talk about what's to come, and they're worried about things that are going to happen a decade from now, 100 years from now, and which is all true, and the numbers get more scary and certainly more impressive. But the thing to remember is this is a $900 million a year problem this year. Next year, it's going to be a bigger problem. And the year after that, it will be bigger. But right now, it's already a $900 million a year problem. Yes, it will get worse, but $900 million is a lot of money, and that's a recurrent problem. So despite the fact that we have these abatement strategies, they're still giving up creating a $900 million deficit. Now, compounding that problem, our animal breeder and genetic friends have done a great job increasing the rate of animal growth. And when in doing so, they've also increased the rate of metabolic heat production. And so we've seen then core temperatures of pigs actually increase from a typical 37 to I think now it's in the neighborhood of 39. So what's happened then is the window of protection has gotten smaller because it still appears that things are pathologic in the 40-degree range. My interest is in muscle. I got into this because I was interested in the potential for heat stress to damage muscle. But also, what's cool to think about is muscle is 40% of total body mass, give or take. And so, small changes in skeletal muscle metabolism can have big consequences for the organism as a whole. And of course, it's the meat product that we ultimately want to eat. I'll just mention this quickly. Those same heat stresses that are impacting pigs are also impacting people. And so there's a whole host of biomedical consequences that take place because of those same heat stresses. And I'll leave it at that. And if that's something people want to talk about, we can certainly do that. But ultimately, at the end of the day, muscle mass comes down to this idea of this balance between synthesis and degradation. And if synthesis outpaces degradation, you have growth. And if degradation outpaces synthesis, you have a loss of mass. And so the big problem, of course, with heat stress is that you end up with less muscle. So clearly, in this case, we've created a scenario where synthesis is being outpaced by degradation. So we can keep those in our box. And the other thing that's become apparent is the potential for amino acid repartitioning. And by that, I mean amino acids that could be used to support growth are actually used to support some other cellular function. So they're not available to support growth. And ultimately, then, that could lead to less muscle without a change in overall degradation or synthesis. And so some time ago, we actually sat down and we brainstormed different things that could go wrong inside of a muscle cell that could impact degradation or synthesis. And it didn't take us long to come up with our list and to realize that this list of these different cellular things aren't independent. They're all actually related to each other. So for example, if there were mitochondrial dysfunction, the expectation is that could have broad cellular consequences. Now, I want to take a step back and just talk about pig production in the simplest of terms, and that is to say, the food goes into the pig. There it is, the simplest of terms. But then the pig is able to use those nutrients to produce ATP, and it's also able to use those supplied nutrients to provide the actual functional building blocks to support muscle. And so the combination of energy available as well as actual nutritive material is used to produce muscle mass. And then, of course, that can be used to produce a tasty product. But during heat stress, we run into some problems. And the first is the animals come off feed, so there's less actual nutrients available. There's also the possibility, and actually, we know that there's intestinal damage. There's the likelihood then of a decreased capacity for nutrient absorption. And so there's less food, less substance available to produce ATP, but there's also less available from a building block perspective to support muscle growth. And of course, then there's meat quality issues. And so that sets the stage. And I was really interested in this entire cascade and just understanding then how all of these combinations of problems, nutrient restriction, heat, metabolic disruption, even changes in behavior could ultimately then impact skeletal muscle. And so 10 years ago now, we did our first experiment, and the muscle that we were working with is the semitendinosus. So this is a muscle that's part of the ham. And what's cool about this muscle is that it has a very, very oxidative part and a very, very glycolytic part. So you can see the very deep red and you can see the white, and it's from the exact same muscle. So it allows us then to understand better how these different characters, how these fibers behave differently. And what we found was that there was oxidative stress. Let me turn on my pointer here. Let's do that here. So what we found was that there was increased oxidative stress. This little bar tells us in the oxidative portions, the red portion, but not the glycolytic, so the white portion. And we thought this was really interesting. We wanted to know more. What's cool about physiology is that it's dynamic. It's constantly changing. And so while at 24 hours of heat stress, we saw that there was oxidative stress, it begged the question, what else is changing? How did we end up? What are the consequences at the same time point? But also, how do we get here? You don't just wake up and have oxidative stress. There's a whole cascade of things that change to create this problem. And so we wanted to know what happened before and what happens next. Do you stay with oxidative stress? Does it get worse? Are other things changing, or is it resolved? And so that really set the stage for really the last decade's worth of work. And so it allowed us to put together this working model of what we think is happening during heat stress inside of a muscle cell. And so I'll just orient you quickly. This is a mitochondria, and you can see there's nutrients coming in, so carbohydrates and lipids and amino acids are coming in, and coming out is ATP, so the energy that we actually use, a cellular currency to perform tasks. And there's a good balance of free radical production and antioxidant capacity. So we exist in what we call a redox balance. Now, this is called the sarcoplasmic reticulum, and it's really just a place for our purposes where we can store calcium. So we're just going to hide calcium in here. And calcium is a necessary part of really any cell, and in muscle in particular, it's what triggers muscle contraction. And this will be a model of our healthy mitochondrial pool. Now, when the animal is heat stressed, and we think a major problem is that the machinery necessary to move calcium from out in the free in the cell to inside the sarcoplasmic reticulum is damaged. And one of the things that we learned from cell biology is that when calcium is free, bad things happen. It's just one of these rules that seems to take place because there's so many things that are triggered by calcium. And so the cells are very smart, and they realize that if calcium is free, that's deadly now, but we can hide it in the mitochondria. And so in fact, what happens in a number of muscle diseases, and we have good reason to suspect in heat stress also, is that calcium ends up hiding, gets really sequestered deliberately inside the mitochondria. And when that takes place, it interferes with the way the mitochondria function. It can trigger a cellular process called apoptosis, and it also creates free radical injury. So now we have production of free radicals that's greater than the capacity for buffering. And we'll ignore that arrow. And then finally, it also creates the problem of decreased ATP production. So this is our working model. And we know, though, that over time, or rather short term, there's another process called autophagy that's activated, and its job is to actually gather up and degrade these damaged mitochondria. And if you look at heat stress over the short term, say through 6 hours or so, what you find is that there's an initial insult, and by 6 hours, that insult is gone, despite the fact that the animals and the muscles remain hot. And one of the things we see is a pretty prominent activation of this autophagy process. But as you continue heating, for some reason, and we don't have a great answer for it yet, though we think it has to do with redox balance, autophagy fails, and we see an accumulation of mitochondria and a resumption of oxidative stress. So if all of this is true, there should be impaired mitochondrial function. And so we worked with Rob Rhodes at Virginia Tech, and he has some fantastic cell culture models, and he was able to actually show impaired mitochondrial function in his cell culture models. And with the bars that we really care about are these two bars compared to this one. So you can see this was the heat stress one, and it's not as good. It's not working as well. So then we thought, okay, we have a handle on what's broken, and that's the first step in understanding then how to fix it. So we wanted to then develop therapeutic interventions, and the idea was that we could protect the mitochondria, which makes sense to me. We think it's what's damaged, or we could actually try to stimulate this autophagy process. And we decided to move forward with protecting the mitochondria, in part because if we stimulate the autophagy process, it's a degradative process, and we want to facilitate growth. We didn't want to facilitate degradation. So we moved forward with mitoprotection. Now, to do this, we actually used a compound colloquially called MitoQ. And really, we went to mitoq.com and ordered it. This is something that you can get immediately. And what it is simply a mitochondrially targeted antioxidant. And so it has a little sequence on it that when the cell sees it, it delivers it right to the mitochondria. And in fact, this compound is in clinical trial. I think it's like 200 different clinical trials, many related to cardiovascular health. So the idea was it's got a good safety history, it's readily available, and it appears to be efficacious. So let's try it in our heat-stressed pigs. And so yes, we took it and we treated our pigs. And the idea, remember, is that we're protecting the mitochondria. We should be restoring redox balance. And so what I'll do is I'll just show you this figure and I'll just highlight these two bars. So what you're looking at, forget mitoQ. That's these bars out here. We're interested in these two bars most immediately because this is the thermoneutral and this is the heat stress. And if you're saying to yourself, those look the same to me, you are correct. This is a huge problem because the whole basis of our model is that heat stress causes oxidative stress. And so this bar should be way up here. And in fact, it wasn't. And we tried a different measure of oxidative stress. And in fact, so again, this bar should be way up here. And you can see that it's not. So clearly this is a problem. So the fundamental change that we saw 10 years ago is no longer happening. And like any good scientist, we repeated this. And in fact, we got the exact same outcome. So we have a decade's worth of work, countless graduate students, millions of dollars in support from USDA, are based on this idea that heat stress causes oxidative stress. And right now, we can't repeat that. So yeah, this is a huge problem. So I did what anybody does and pouted for a little bit and started to walk around and ask some colleagues and think what might they know, what ideas might they have. And I'm fortunate to be blessed with just some fantastic colleagues at Iowa State. And one of them suggested that we actually consider the biological sex of the pigs. And so we did. And it happened by chance. All of our old work had been done in gilts and was only those most recent two studies that had been done in barrows. And so we wanted to know then, could it actually be something as simple as a difference in biological sex? Now, I am a hoarder. I have an incredible collection of ice packs that we get in the mail and cardboard boxes. But I also have a good collection of tissues that we've used from previous experiments. And we're able to dive into our freezers and actually find the original tissues, just a little bit left of the original tissues that we had worked on so long ago. And so what I'll do is just orient you quickly. So this is oxidative stress, an oxidative stress measure for the thermoneutral females and the heat stress females. And you can see they're elevated. And this is from the thermoneutral males and the heat stress males. And you can see there's not much of a change here at all. And so it at least supported the idea that, yeah, there could be an effective biological sex on heat stress mediated outcomes, including oxidative stress. So it supported this hypothesis that males would be more resistant to heat stress mediated dysfunction than females. And so we wanted to address this directly. So to do that, we had some thermoneutral males and females, so barrows and gilts. And then we just maintained them for a week or so under thermoneutral conditions. And then we had some barrows and gilts that we heated for 24 hours or we heated for 7 days. Now, I want you to keep in mind, these are the same genetics, the same truckload of pigs, and they were in the same room at the exact same time. And within that room, we actually alternated them barrow gilt, barrow gilt, barrow gilt down the alley so that we mixed them as best as we possibly could. So at the end of the day, what we figured out is that, and so these are our heat stress animals, and these are the heat stress gilts. And so yes, we can make animals hot. We're good at that. But what's interesting is that the heat stressed gilts, the heat stressed females, were actually cooler than the barrows, despite the fact that they were in the same room at the same time. And you see that taking place on day 6 and day 7, and they were also then cooler than their initial 24-hour time point. That finding was completely surprising, but was also supported by muscle temperature. And before I get into those data, I just want to orient you to this figure, because you're going to see a number of figures that look something like this. So it will always be the treatment on the bottom. The barrows will always have the blue bars, and the gilts will always be the solid bars. And we made them more intensely red as you get hotter. Capital letters will always refer to differences within barrows, and lowercase letters will always refer to differences within gilts. And then finally, if there's an asterisk above a bar, it indicates a difference between sexes within a single time point. So in this case, you can see that yes, muscle temperature increased following 24 hours of heat stress, and it increased fairly uniformly in barrows and gilts. But what's interesting is by 7 days of heat stress, both barrows and gilts were able to decrease muscle temperature, albeit it was greater on the gilt side, which is fairly consistent with what we're seeing with core temperatures. Both barrows and gilts decreased feed intake, as you would expect with heat stress, and that reduction was fairly uniform. But what we're really interested in was this idea of feed efficiency. And so we used gain to feed as that measure. And again, our hypothesis here was that barrows would be more resistant to the negative consequences of heat stress and should better maintain a feed efficiency. So what you can see is that, yes, barrows and gilts both decreased feed efficiency to actually negative, so they're eating food but losing weight. Unfortunately, though, they weren't quite statistically different. And the reason is because these two animals right here, these two gilts, somehow maintained a really, really high feed efficiency. I can't really explain that to you well, except to say they're not quite outliers, and so there's no reason to discard them. But we did note that the variability around this time point was really high, and in fact was greater in gilts than it was in barrows. And so we performed that measurement. So we calculated the variance coefficient, and in fact, yes, you can see it's significantly different between the gilts and the barrows. And so while I can't tell you the barrows are more efficient than the gilts following 1 day of heat stress, what I can tell you is that within a population, you're more likely to find gilts that are more negatively impacted than barrows. Despite being off feed for essentially 7 days, blood glucose was largely maintained, albeit this is a significant reduction in barrows and gilts. The effect size is actually fairly small. NEFAs, so fatty acid, one would expect when you come off feed, you would mobilize fatty acids. And while again, we do see some changes here in barrows and gilts, the magnitude of those changes is actually really small. And if we had included a pair-fed thermoneutral group, it would actually put in context exactly how small that is. But it's I think it is a third of what you would expect with a pair-fed thermoneutral group. So yes, we're kind of mobilizing fatty acids, but not near to the extent that one would expect. So animals come off feed. The expectation is insulin should decrease, and that's roughly what we see. But glucagon is a hormone whose job it is to help maintain blood glucose. And you can see glucagon also comes down fairly similarly in barrows and gilts. Cortisol also declines, and cortisol is one you would expect to help maintain blood glucose. And T3 and T4 are interesting. These help govern basal metabolic rate, and the expectation is that those should both decline with heat stress, and they do. So this is something that we've seen before, a reduction in T3 and an immediate reduction in T4, and then a slight recovery. So just to refresh, it looks as though there are differences in barrows and gilts with growth efficiency, particularly during that 24-hour time point. Somehow blood glucose is being maintained, and that's not exactly clear, but it looks as though there's metabolic differences, at least at the phenotypic level, between how barrows and gilts are responding to heat. And again, this is despite the barrows are actually warmer than gilts, which is pretty cool to think about. So we wanted to then get into mitochondrial function. And so this is data that Sarah White Springer helped produce for us. She's a fantastic collaborator at Texas A&M University and comes up and performs some really nuanced measures with mitochondrial function. And I won't get into all the details here, but what I'll give you the take-home message, and that simply is that there are differences with the way barrows and gilts are responding to heat stress. This is complex 1 and this is complex 2 of the mitochondria. You can see there's differences. The take-home message is that gilts are relying on complex 1 for ATP production, which is interesting because it actually promotes free radical injury and free radical leak, whereas barrows appear to be relying more heavily on complex 2, which is more resistant to electron leak. So it looks as though the animals are behaving differently with heat stress from a growth efficiency perspective. The mitochondria, we can now say objectively, are operating differently between barrows and gilts during heat stress. We wanted to then better understand how that might actually impact the metabolome. So this is just an example of the data we produced from a metabolomics analysis. So this just gives us wholesale what metabolites are around. And then from those, we can actually then start to understand how the TCA cycle is functioning, glycolysis, beta oxidation, just different pathways that are important for cellular metabolism. But I just want you to see the picture. This is what thermoneutral gilts look like, and this is from muscle from thermoneutral barrows. And you can see under thermoneutral conditions, barrows and gilts are actually pretty similar. Not identical, but pretty similar. And as you advance through to heat stress, these are 24-hour gilts, and you can see these look wildly different than the thermoneutral gilts, and they also look wildly different than the thermoneutral barrows. And so it tells us that, again, it's these barrows, these animals, sorry, the gilts are behaving in a different way metabolically than are the barrows following 24 hours of heat stress. And as we continue on through 7 days, we see more uniformity between barrows and gilts that differ substantially from the thermoneutral condition. But again, during this chaotic period, particularly during the first 24 hours, we're seeing big differences between the two sexes. So we wanted to understand then, what does mitochondrial structure actually look like? So we see that the function is different, the consequences of their activity is different. We see the differences in metabolome, but what do they actually look like? How might their structure then be related to their function? And so what you're looking at are electron micrographs of skeletal muscles. So this is zoomed in something like 1,000-fold, like an insane degree. And this is what thermoneutral muscle looks like in gilts and in barrows. And it's beautiful. You can see how highly organized the structure is. It's repeatable. So this, just as a muscle biology lesson, because I think it's just so interesting, this from here to here is called a sarcomere, and it's the contractile unit of muscle. And when muscle contracts, this black line moves closer to this black line, and they move toward the middle of the sarcomere. So both ends move in to what we call the M line. And you can see the M line in the middle. But you can see how highly organized it is, even moving between what we call myofibrils. Very, very highly organized. And the same thing is true in the muscle of barrows. As we apply heat for 24 hours, you can still see a fair degree of organization. We don't see widespread damage, but these large structures here are the mitochondria. They've entered a hyperfused state. And we saw these fairly routinely in the gilts, but we never saw them in the barrows. The barrows maintain a normal mitochondrial structure, and they were also just not massive. These are running anywhere from 1.5 to 2 sarcomeres in length, just to give you some scale, whereas these average somewhere in the neighborhood of less than 1. As we continued heating through 7 days, we no longer saw accumulation of mitochondria in the gilts, but we did start to see damage to the sarcomeres. So you can see this looks ripped and this looks damaged, and there's a bunch of debris in the middle. We did see that in the barrows as well, but to a lesser extent. And so now we have good evidence that at the phenotypic level, there's differences in barrows and gilts in the way that they're responding to heat stress. At the mitochondrial level, there's differences. The metabolome is different. And now we can actually see mitochondrial structure differs between barrows and gilts. So it gives us a lot of confidence that these animals, that biological sex does mediate quite a bit of heat stress mediated biology, but not all of it. And that subtlety, I think, is important. But nevertheless, there's certainly differences in the underlying mechanisms in the way these animals are responding to heat. But we also wanted to get into this idea of protein synthesis, because ultimately, right, from a producer perspective, we still want to produce as much meat as we can for as little inputs as we can, which comes down to a balance between synthesis and degradation. So I had it in my head that heat stress would cause increased degradation. And in fact, I think a lot of people say that, and it's common to hear people say it increases muscle breakdown. Muscle breakdown is essentially breaking proteins into their component amino acids, and those amino acids then are used for some other function. So we started with assessment of proteolysis. And I was pretty shocked to find that headline says it all. Heat stress does not promote proteolysis. And what you're looking at are measures, some biochemical indices of proteolysis. MRF1 and MAFBOX are 2 proteins that are frequently elevated with muscle atrophy or muscle wasting conditions. They absolutely did not change. Then we actually measured the proteases themselves. So these are the things that are actually responsible for chewing big proteins and making them smaller, essentially amino acids. You can see that calpain activity absolutely is not increased, and you could even argue that there may be a reduction. And the same thing is true of proteasome activity. So calpains are essentially, you can think of them as scissors. So they take big polypeptides, big proteins, and make them the smaller polypeptides. And the proteasome's job is essentially, it's like a barrel, and it's like a wood chipper, and it just takes in what the calpains give it and just spit out amino acids. Absolutely not increased. And so it really begged the question, does heat stress actually increase proteolysis? And at this point, we're not thinking the answer is yes. And it actually turned out those data fit pretty well with an ongoing project that we've since published in heat stress dairy cattle. And in fact, we found very similar things, that heat stress, in fact, did not drive proteolysis in skeletal muscle from dairy cattle. So the answer doesn't appear to be degradation. And so we wondered then, might it actually be impairing synthesis? So we did some measuring synthesis is actually exceedingly difficult, but it's one that can be done pretty easily by measuring markers of synthesis. So in fact, that's what we did. So ribosomal capacity, ribosomes take mRNA and actually build proteins. So if you have a larger ribosomal capacity, your capacity to produce proteins is greater. And if you have a decreased ribosomal capacity, again, your capacity to produce proteins is diminished. And you can see over time that appears to be the case. It's similar between barrows and gilts, but definitely it appears to be a negative impact on ribosomal capacity. And these are also measures that get to ribosomal capacity and ribosomal abundance. All of them are headed in the same direction. So these appear to be headed downward. We wanted to get into some signaling. So mTOR is a very common driver of protein synthesis. And you can see, surprisingly, the activated form appears to be increased at least at 24 hours in gilts, though not by 7 days, which was interesting. And so we actually followed that up and we measured downstream, is it able to pass that phosphate group along and actually activate its binding partners? And what we found was that was not the case. And in fact, there's a reduction in phosphorylated 4-EBP in gilts as heat stress continues. And the reason this one matters so much is because 4-EBP, this group, is actually an inhibitor of protein synthesis. The phosphorylated form is turned off. So this is how you turn off the inhibitor. And you can see that in gilts in particular, there's a greater turning off of the inhibitor. So the expectation is decreased ability for protein synthesis. So all of these data are indicating the same thing, that protein degradation appears not to be impacted at all. And really, if there's losses in muscle mass, it's because of a synthesis issue. And then we started thinking, well, if we're decreasing protein synthesis, that might free up amino acids then that could support the amino acid pool in blood and allow this pig then to provide increased stress resistance, or maybe you're mounting an inflammatory response, because we know that heat stress is pro-inflammatory. So maybe these amino acids then are being used for some other function. And so we started with just some simple measurements. And what we did is we measured the amino acids in blood, and then we measured the same amino acids in muscle. And so what you're looking at is called a bubble plot. And what we care about is the size of the circles. And so we've quantified all of these, and we've actually performed our statistics, etc. And what you'll see if you look over these essential amino acids, what you'll see is that at the end of the day, there's really not a lot of differences between barrows and gilts, though there are definitely some. The circles end up a lot smaller following 24 hours and 7 days of heat stress than they do under thermoneutral conditions in blood. And in fact, that's exactly what happens. However, in muscle, if you really start looking at these, most of these actually stay just about the same size. And so on the whole, essential amino acids are preserved in muscle, though they decline in blood. And we did the same measurement for non-essential amino acids. And what you find is that in blood, these things come plummeting down at 24 hours, really not a big change after 24 hours, but in muscle, they're largely preserved. So when you add them all up, this is roughly what you find. So in blood, there's about 30%, 40% reduction in circulating amino acids. Fine. In muscle, this appears to be largely preserved. And yes, there's a small reduction in barrows, but on the whole, the reduction that you see in blood far surpasses what you see in muscle. And then we started thinking about then other ways that you can categorize amino acids, particularly in regard to metabolism. And I'll just spare you the details, but I will tell you that we considered gluconeogenic amino acids, and those were absolutely preserved in skeletal muscle, though they came plummeting down in blood. And it wasn't until we considered separately the ketogenic amino acids, lysine and leucine, that we saw changes in muscle that more closely paralleled changes in blood. And so it appears as though ketogenic amino acids are being selectively released from skeletal muscle. And that's roughly where we are now. And so we're considering then what to do best, what to do with that information. So I will give you my one slide summary. Heat stress certainly negatively impacts agricultural production, and yes, it also impacts human health. We hypothesized that barrows would be more resistant to heat stress than females, and ultimately what we discovered is that females, yes, were able to cool themselves biologically to a greater extent than the barrows, but despite that reduction in temperature, growth efficiency was not improved, and if anything, you would argue that growth efficiency was more negatively impacted in the barrows and gilts than in barrows. We saw changes then in metabolism. Then we started with thinking about how the animals are growing. Mitochondrial function was different. The metabolome was different. Even mitochondrial structure was different. We also considered this idea of proteostasis, so the balance of synthesis and degradation. It appears as though protein degradation is similar, but protein synthesis is decreased in barrows and gilts. And we're certainly interested in this possibility that animals are actually ketotic during heat stress. As far as what do we do with this, at this point, I would say a little more work here before we make some big changes. But we're certainly interested in this idea that there may actually be differences that we should be considering from a therapeutic perspective between barrows and gilts if the underlying mechanisms and underlying injury caused by heat stress differs between barrows and gilts. It would make sense to apply a solution to barrows that gilts don't have, a problem gilts don't have, and vice versa. We're also interested in this idea then, if that's the way that we need to go, what does that mean then from a management perspective and thinking about this from a pig systems perspective? We have no idea how long these differences last. For example, if, say, an animal were heat stressed for a week, what happens in the weeks that follow? We think that's pretty important because, one, the injury may change. The injury may differ between barrows and gilts. And also from a practical perspective, if you can apply an intervention, you need to know when to stop. And so that's work that we think is absolutely essential as a next step. And the other thing that we're getting into is this idea that other organs are also impacted by heat stress. And I have a very talented grad student, Missy Rhodes, who's begun to explore this and has nicely shown heat stress damages these hearts. And it's really now getting into the mechanism as to why. And there's some cool data now coming out of sows that indicates that there's some increased heart damage, and some of these animals may be dying because of heat-related cardiac injuries. And one of the things that we have is some cool preliminary data showing just 3 months following a 3-day heat exposure, there was still apparent cardiac injury. So we're certainly interested in that. And the other thing that she's gotten into are the kidneys and better understanding how heat stress might be damaging the kidneys. Well, really asking the fundamental question, does heat stress damage the kidneys? The answer is a resounding yes. And now we're getting into exactly why and how. So as I said, Tori is a grad student that led most of this work. Missy Rose is contributing and supported Tori in some of this initial work, and she's doing some great stuff on her own with hearts and kidneys. And the group here, I've been fortunate to have some great people in my lab over the years, and also just some fantastic collaborators. Lance Baumgart and Rob Rhodes have been kind of brought me into this heat stress field more than a decade ago, and Sarah White is a new collaborator from A&M. I've been fortunate to be supported by USDA for some time. And I'd be happy to answer any questions.
Thank you, Josh, for that very good presentation. It certainly gives us a lot to think about. I kind of feel a little bit overwhelmed at times because there's so much that happens to that animal, but yet we still don't fully understand it. Right. And I'm always the person that wants to fix it. So I'm still kind of like, what do I do next? But we'll stay tuned because I'm hoping you guys can tell us how to fix that.
Yes. Yeah, understanding the problem is that's that first step, and it's such an integrative injury that takes place.
Yes, yeah. I mean, I worked off of the concept for years, oh, they need more energy, right? But we didn't really think about the amino acid piece. And so I think this is really intriguing, and certainly we've always had conversations around in a production barn, oh, well, the barrows look to die faster than the gilts or sooner than the gilts. And so, it's kind of fun to see maybe some of that explanation as to why come around. For those of you who are on the call, please feel free to drop any questions you have in the chat. We do have a question for you, Josh. So the question is twofold. Is there any work on chemically castrated pigs? And if not, if you had to hypothesize, would these be similar to gilts or barrows?
Awesome question. In fact, I did not show these data. I took them out, and maybe I should have shown them. We picked up testosterone on one of our measures. So we did a panel measurement, and testosterone came out. In fact, the gilts had greater testosterone than the barrows. And I think that's going to turn out to be a pretty important part of the story. So to answer the question, my expectation is that chemically castrated barrows would respond in a similar fashion as the actually castrated barrows. But I would like to use the chemically castrated barrows as a control group in some future work. Just one, so we can demonstrate that empirically, but also I just think it's an important control moving forward.
Mm-hmm. Yeah, that'd actually be quite interesting, right? Because if they're a boar, are they better than the gilt then if they're carrying more testosterone? But then if we give the second injection, then are they more like the
So
barrow?
I think it would be the opposite because it's the gilts that
The
are more
opposite.
susceptible. So
Okay.
as you have testosterone, I think that's gonna pre-expose you to an increased susceptibility to thermic injuries. And as you look at human populations, a lot of times it appears to be anyway, males being more negatively impacted than females under the same heating conditions.
Mm-hmm. Mm-hmm. Very interesting. Okay. So that would be, yeah. So timing, right? Timing will be important to when animals will be
Yes.
exposed to heat. Yeah.
100%. And then timing is also important to think about cyclicity for females. So I went over it quickly, but we're using animals that are about 3 months of age. Just we wanted to simplify things as best we could. As we get into cycle with females and get into sows, how that impacts heat stress mediated outcomes is a whole new complexity that we're eager to consider.
Mm-hmm. Yeah, I think the other one I have for you, because we've talked about it in the past, is full feed versus control feeding. And I know
Yep.
obviously with the 3-month-old animal, we're allowing full feed, full access, but would we see a different outcome if they are control fed versus full fed?
Are you talking about the control group or the heat stressed animals?
I'm talking about heat stressed animals.
Yeah. I don't know because already they're decreasing food consumption by about 50%.
Mm-hmm.
You can start to play with the components of that feed. And one of the things that, in fact, next grant that goes in is actually going to be to consider the idea of lysine and leucine loading and how might that actually impact a diet and impact these outcomes. But beyond that, I'm not sure.
Yeah. Or even in the recovery phase, right? So after a heat stress
Mm,
event, is it better,
yes.
right? One or the other.
No idea, because I worry about the intestine and I worry about
Right.
this idea of lingering intestinal damage. And if you're pushing more food through it, is that is that gonna make things worse? Or is increased nutrient availability better? I don't know.
I don't know, right? Because as nutritionists, we always think about, oh, let's just, right, the faster we can get them recovered, the better off we are. But that may not biologically make sense, right? There might be, and the body's really good at protecting itself, but there might be some
Yes.
other reasons why it might be a different mindset. Right?
Yeah. So one of the subtleties that came out of this work is we actually considered nutrient partitioning within the GI tract or feed partitioning within the GI tract. And what we found during heat stress was that the animals actually, so the food is eaten, and that's when we measure it. So how much food disappears from the dish or from the bucket or the trough, and we count that as food consumed, which is true, but that doesn't mean that it's nutrients absorbed. And what we figured out is that these animals are sequestering food largely in their stomachs. And it looks as though then the transit through the small intestine is more rapid than it is under thermoneutral conditions. And
Mm.
I don't know what to do with that information except to say and highlight that we've got to expose the feed to the small intestine to enable absorption. And if it's trapped in the stomach, um, you need all you want, but the absorption isn't taking place. And
Yeah.
I don't know how long that lasts, and I don't know how long that lasts during recovery. So
Hmm.
if something like that continues, the animal's eating a lot, but it's going to be on the back end, there's going to be a lot of valuable nutrients that are lost.
Hmm. Yeah, I always think about, if it's a really hot day and then the night it cools off and the pigs even go and they just eat a large amount of feed, right? So I think that kind of goes back to some of that conversation. But then if we get the cyclicity of it, of hot day, hot day, cool night,
Yeah.
what does that even do for overall recovery?
Yeah.
Um,
And I don't know. I don't know. These are great questions because we do
yeah,
know that even the hot day, that time period can be damaging.
right, right. Yeah. Well, lots of lots of work ahead of you, Josh, but it
That's right.
definitely was very interesting. So we do want to thank you again for being on today with the IPIC team. And for those of you that are listening or watching, this was Dr. Josh Selsby, who's with the Iowa State College of Agriculture and, of course, the Department of Animal Science. So thank you again, Josh, for your time. We greatly appreciate it.
Thanks for having me.
Yep. Have a great day.